Hi guys, let's dive right into chapter 12. We are going to be doing section 1 in this video, which talks about the genetic material. We're going to start out talking about a bunch of different types of experiments that scientists conducted in the mid-1900s. There's a lot of information out there about those experiments, so I'm going to link a couple of Khan Academy videos into Google Classroom so that you guys can watch those to gain more information about it, because they do a much better job explaining the experiments than I can in this video. So if you want to learn more, those videos will be posted at the same time as this one. The first experiment we're going to talk about is Griffith's experiment. He had used the strep bacteria. He used 2 different strains. Some of them he treated with heat, some of them he didn't. Some of them had genes for a capsule, a mucus capsule, on the outside of the bacteria to protect it. And he did a bunch of experiments with that. Further research was done by Avery and his team. So at this point, scientists wanted to know whether DNA or proteins were the hereditary material. They knew that both DNA and proteins were important, but they didn't know which one did which thing. They knew that proteins were made up of 20 different amino acids, and they knew that DNA and RNA only contain 4 different types of nucleotides. So they figured out what the genetic material had to be able to do. It must be able to store genetic material, genetic information. It must be stable and able to be replicated accurately and passed on from generation to generation, but also able to undergo mutations that provide genetic variability. And scientists did all kinds of experiments to figure out that DNA is the one that can fulfill all of those functions. So Griffith's transformation experiment, this is the one we talked about on the previous slide. Mice were injected with 2 different strains of strep. One of them was encapsulated and one of them was not. The S strain was virulent and the mice died. Okay, the R strain was not virulent and the mice lived. Okay, the R strain did not have an outside mucus capsule. The S strain, however, did. So here is a diagram that is in your textbook showing how some of the bacteria was treated with heat and some of it wasn't, and the results of those experiments. Avery, MacLeod, and McCarty's experiments, scientists argued that DNA lacked variability to be able to store genetic material. Avery and his colleagues used enzymes to break down DNA. They used DNase to break down DNA. They used RNase to break down RNA, and protease to break down protein in separate experiments to digest the substance, which allowed strep to produce the capsule around it and become virulent. The only enzyme that had an effect on it was the DNase, which prevented the transformation from occurring, which basically stopped the bacteria from making that outside capsule. So this proved that the DNA was the transforming substance. It was the thing that was passing on the DNA. Okay, DNA was being passed on, and that was allowing them to produce this outer slime capsule that made them better protected. Some other experiments named Hershey and Chase, they used radioactively labeled DNA in E. coli, and they discovered that radioactively labeled DNA, but not protein, ended up inside the bacterial cells, causing them to be transformed. So the viruses, they were able to transfer the radioactive DNA, but they weren't able to pass on the radioactive protein. So only the genetic material could have caused the transformation, could have caused the radioactive DNA to be inside. So this experiment showed that only DNA and not protein was the genetic material. So transformations occurring in organisms today. Scientists are working on genetically modifying organisms, also called GMOs. You may have noticed on a lot of food labels, some of them say non-GMO. Well, that just means that it is an organism that has not had its DNA altered by scientists. Scientists are taking genes from other organisms and putting them into organisms that would provide them with a competitive edge, make them able to withstand heat or flood or drought or pests better than organisms that are not genetically modified. Scientists are doing this mainly in agriculture and commercially grown crops to make them more productive so that they don't have to use as many pesticides or herbicides, so that they don't have to water as much, so that they basically get more bang for their buck. Okay, scientists are using green fluorescent protein as a marker. So they put that into an organism because it's easy for them to make sure that it ended up where they wanted it to. The jellyfish, it comes from jellyfish. The jellyfish gene is isolated and then transferred to a bacteria or the embryo of a plant, pig, or mouse. When the gene is transferred to another organism, the organism will glow in the dark. Scientists have actually created glow-in-the-dark cats. It's pretty cool. If you have time, Google it. It's really interesting. So scientists are able to take DNA from one organism and put it into a different one. All right. So people have their own opinions about whether that's good or bad. If you guys want, we can have a discussion about this in class tomorrow. So here is a diagram. Okay, so here we have the DNA. Okay, here we have the capsid, which is like a capsule outside the RNA that holds it all in, and it's basically injecting the DNA into the bacteria. Okay, so here we have the virus injecting a copy of DNA into the bacteria. Okay, so the virus, non-living, but it's injecting DNA into the living bacteria cell. DNA contains two types of nucleotides, purines and pyrimidines. Purines are made up of adenine and guanine, represented A and G respectively, and pyrimidines, which are thymine and cytosine, which are represented T and C respectively. A scientist named Chargaff discovered that there are constant amounts of DNA of each of those. He noticed that there's always the same amount of adenine and thymine in a strand of DNA, and there's always the same amount of guanine and cytosine in DNA. So they are constant in the DNA. That led him to believe that adenine bonds with thymine and cytosine with guanine. Okay, and this suggested that DNA uses something called complementary base pairing to store genetic material. So whenever DNA is replicated, basically it breaks right down the middle, and you can easily build another strand by just looking at what's already there. And if you see an A for adenine, you automatically know that the other side of the DNA strand is going to have a T for thymine. Each human chromosome contains, on average, 140 million base pairs, which means 140 million adenine, cytosine, guanine, thymine. Okay? And here is a picture of what those molecules actually look like. Every single one of them contains a phosphate molecule, which is this P with the O's and the H's, bonded to a deoxyribose molecule. Okay, that's a sugar molecule, and then it bonds to a nitrogen base, which is either the adenine, the guanine, the thymine, or the cytosine. Now, the amount of each one varies from species to species. So in humans, roughly 31% is adenine, roughly 31% is thymine. There's a little bit more thymine because in the processing of RNA, it'll add thymine at the end to protect it, which we'll talk about later on this year. But you'll notice that corn only has about 25-26% adenine in it. Okay, so roughly the same percentage in all humans of each of those nitrogen bases, Okay, roughly the same amount of each of those nitrogen bases in corn, but whenever we compare species, it's a little bit different. X-ray diffraction. This amazingly awesome woman named Rosalind Franklin studied the structure of DNA using X-rays, and she found that if she concentrated a viscous solution of DNA, it can be separated into fibers, and then she could take an X-ray of it and figure out the shape of the DNA. So she figured out that DNA is in the form of a double helix, and that some portions of that helix is repeated. And so eventually a colleague showed Franklin's work to two guys named Watson and Crick, and they used that information to publish their own paper describing the structure of DNA. So here is the famous, there we go. There's the famous image that Rosalind Franklin took that helped her figure out that DNA is actually a double helix. So that was shown to Watson and Crick without her knowledge. So Watson and Crick, they took information from a bunch of other scientists and they figured out that it's a double helix, similar to a twisted ladder, and that it has a sugar phosphate backbone that make up the sides of it, kind of like the sides of a ladder, and that hydrogen-bonded nitrogen bases make up the rungs of the ladder. They also used other people's research to discover that they were anti-parallel, meaning they were side by side, but they were facing different directions. And they discovered that in their model, DNA must be read in the 5' to 3' direction. That has to do with the way that the sugar molecule is pointing. And so it's replicated in that 5' to 3' direction, which we'll talk about more later. Complementary base pairing ensures that a purine is always bonded to a pyrimidine. Okay, it has to do with the number of hydrogen bonds that are formed. So A always bonds with T, and G always bonds with C. They fit together like a puzzle. And in 1962, Watson and Crick received a Nobel Prize for their, quote, discovery. At that point, Rosalind Franklin had died, so she was unable to receive the Nobel Prize because they are not awarded after death. So here's a picture of Watson and Crick's DNA model. The sugar phosphate backbone is here. This is our sugar. Here's our phosphate. Okay, and in the middle we have the nitrogen bases. See that cytosine always bonds with guanine and thymine always bonds with adenine. And what holds them together is the hydrogen bonds. And that's it for this video, guys. If you have any questions, let me know.